So the topic that we will be covering today is about the replication of bacterial DNA. So we're going to learn how bacterial cells copy their DNA when they need to procreate. And I want to make very clear distinction between the process of DNA replication and the process of gene expression. Often, these two processes are mixed up. They are different, okay? Please make sure that you understand. Replication has nothing to do with the expression of genes. So, in the process of replication, one DNA molecule is doubled to produce two DNA molecules, one for each daughter cell. Does that make sense to you? Okay. Now, when the whole field of molecular biology just started to develop, there was really a big question: how replication of DNA occurs? Because there are, well, at that time, scientists were thinking about several possibilities, and I wanted to highlight two of them. So first possibility was that you have an original DNA, and then after that replication, you have an original DNA, you have a completely new copy. So this mechanism of replication was called a conservative. And another possibility was that each strand of DNA was copied independently. So what you would end up with is something like this. Strands would be separated, and each strand would be copied. Does that make sense? You see that? So this process right here was called semi-conservative replication. And the main question at the time was, which of these mechanisms actually occurs? And I give this example. I don't want to confuse you. Now, look at this. Both of these mechanisms, if you don't know how the process works, both are equally conceivable. And scientists said, we don't really know which one it is. So what we're going to do, we're going to set up an experiment. And they predicted results of this experiment if the mechanism will be like this, and the results if mechanism would be like this. Okay, so they basically said, if DNA is replicated this way, we're going to see A. If DNA is replicated this way, we're going to see B. Am I clear? That's the example of perfect scientific thinking, scientific method. You're not biased about the possible mechanism. You just say, that's what can happen. Let's check. So to make a long story short, they did an experiment, and the experiment demonstrated very convincingly that semi-conservative is a way to go, and this one is not correct. It was just such an elegant, an elegant approach to science, When you don't expect the result, you don't know the result. Am I clear? Okay, great, fantastic. So we basically figured out that to replicate the DNA, you need to separate two strands and copy each of the strands in the complementary manner. Does that make sense? So here is when complementarity sort of comes into play. Okay, now look at this thing here. Let's say we're going to name this strand number 1 and this strand number 2. So if this is number 1 and this is number 2, remember these strands are complementary. You see what I'm saying? DNA is copied based on an ATGC pairing. So if this is strand number 1, then the newly synthesized red strand will be a copy of number 2. And if this is a strand number 2, the newly synthesized red strand will be strand number 1. Okay? Does that make sense? Just, and what you end up with, look at this. You end up with 2 copies of the original double-stranded molecule. We clear? Now, our next probably 20, 30 minutes will be dedicated to the discussion of the whole process of DNA replication. Okay? That makes sense. So there's a list of enzymes in the lecture notes with their main functions. So you do need to know the functions of the enzymes. I will ask you to match the name and the function. And also you have to have a general idea of the whole replication process. The picture that you have in your lecture notes, the picture of the DNA replication, is a static snapshot. Meaning that what you're looking at is a moment in DNA replication. It does not really reflect the process. I cannot draw the process, I cannot do the animation on the whiteboard, but at least I can try and do step-by-step process, to kind of illustrate what is going on during the DNA replication, okay? So we're going to be talking about replication in bacteria, which means single circular chromosome, okay? Not many chromosomes like in the eukaryotes. And we're going to start with the fact that when we say that chromosome is circular, you have to understand it's not a perfect ring, okay? Imagine that you have a closed loop, but this loop is all coiled. My best analogy to coiling of DNA, ever used the phone with a wire, okay? You know this spiral wire, how it all gets coiled around and wrapped around itself? DNA does the same. It's called supercoiling. Does that make sense? DNA is supercoiled, and before you can actually separate the strands, start copying them, you need to straighten it up. So the protein that straightens up supercoiled DNA molecule is called DNA gyrase. Okay. DNA gyrase or topoisomerase 2. And all it does is straightens up the DNA. So we've got straightened DNA. Okay? Next thing to do is to unzip the DNA molecule. Does that make sense? Separate it. Now here, I'm going to put a protein that unzips. Come on in. Oh, okay. Next we are going to put another enzyme, which is called a helicase. So what helicase does is unzips the DNA. Okay, now I want to pause here and kind of explain a little bit more. The best analogy for what helicase does is a zipper like the one on my backpack. Does that make sense? Helicase is like that little thing on the zipper that you're moving and pocket becomes open, right? Now, you don't have to walk to my backpack to see that on each zipper, this particular one has two running things. Does that make sense? I bet yours have the same. Well, not all of them, but yeah, Nanishka's bag has two zippers. You can put them to the sides and they will open it, right? Make sense? So if you think about the DNA molecule, there's no reason to unzip it only one way. Does that make sense? So it will be unzipped both ways. So you're going to have helicase here, and you're going to have gyrase. So far, we're good? Now, you look at this, obviously, what I show you here is also kind of a snapshot. So there should be some location in the DNA where initial separation starts. Does that make sense? You understand that? Like, where are you going to start splitting those strands? That start, the replication, is called origin of replication. Here. Origin of replication. Which pairs of nucleotides form weaker complementary pairs? And so origin of replication is a lot of AT pairs, so it's easier to separate. Does that make sense? Okay, cool. Look what we've got. Now, here's the thing. I'm going to be focusing on the left replication fork, but keep in mind, same events going on the right replication fork. So let's put the directions. This replication fork is moving to the left. This replication fork is moving to the right. You good so far? Okay. And finally, I want to put the directions of the DNA. So 5 prime, 3 prime, 5 prime, 4. Does anyone remember in which direction the DNA molecule is read? 3 to 5. So if we start here, starting point. We start here, we can read DNA into this direction, right? Let's not talk about this strand yet. Let's focus on that one. So first of all, when you separate these strands, they immediately, quote unquote, want to come back together. They want to renew. To prevent this, protein called single strand binding protein binds to each individual strand and prevents them from coming back together. Does that make sense? Now everything is ready for the enzyme called DNA polymerase 3 to start making a new DNA molecule. Here's one little conundrum. DNA polymerase 3 cannot just bind to the DNA and start copying. It needs a special structure that's called a primer. Okay? In my world of imperfect analogies, primer is the starting line for a marathoner, okay? The person wouldn't just walk outside and would run. The person needs like this massive thing, like here's the start. That's where you're supposed to start running. Does that make sense? So what is a primer? Primer is RNA about 20 nucleotides long. And I want to emphasize this. It's RNA, not DNA. Does that make sense? So primer gives the DNA polymerase a site where it is supposed to start copying. We good? So I'm going to draw DNA polymerase in sort of dashed appearance because it's kind of moving. Okay, so look at this. So it originally was here, but now DNA polymerase is here. Okay, and while it was moving, and it is moving with the fork. You see what I'm saying? It is moving with the fork. As the fork moves to the left, DNA polymerase moves to the left. Let's call it polymerase. Okay, and it's number 3. And what it does while it is moving, it makes a new DNA. Does that make sense to you? So one more time. RNA primer is synthesized. By the way, let's put down which enzyme does it. Enzyme called primase. Synthesized by primase. Okay. So primers puts the primer in place, DNA polymerase 3 binds to the primer and starts the synthesis of the new DNA strand. Does that make sense to you? Okay, good. There was something I wanted to mention. Oh yeah, to clear my consciousness, you may read in some of the books, you may get a perception that DNA polymerase 3, when it replicates DNA, it must have a primer. Well, DNA polymerase 3 definitely must. But there are some DNA polymerases, some, that can replicate DNA without a primer. They will not be on the exam. This is the part that you don't need to know. I just must tell you this because, you know, that's true. You good? Moving on? Okay. Here's my question. And I promised before, if I ask you a hard question, I'm going to tell you. So this is not a hard question. DNA polymerase 3, here. As the fork moves to the left, and DNA and this whole construction moves to the left, like a zipper. Does it have to experience any interruptions when it is reading DNA? Grace? No, because it's 3' to 5', right? It's good. Now, obviously, in order to make replication more efficient, we want to put DNA polymerase on this side as well. Can we put DNA polymerase so it will be reading DNA? Why? It will be 5 to 3, which is not possible, right? So how can we get around this problem? By the way, this strand that is synthesized in uninterrupted fashion is called leading strand. Now, let me see if I have something. Oh yeah, perfect. I have a book here. Okay. So how I normally would read the book. I would read it like this, right? from the beginning to the end. Okay, that's our leading strand. In this case, basically, I need to read bulk from the end to the beginning. You see what I'm saying? How would I approach this? So I would find chapter 24, and we'll read chapter 24 as I would normally do, right? And then I'd go back to chapter 23 and read chapter 23. And then I would go to chapter 22, 21. So I would be going backwards, but I won't be reading backwards. Does that make sense? So this is what happens on another strand. This will be somewhat of a process drawing. So the primase makes the primer, okay? And DNA polymerase 3 binds to it, moves forward, synthesizing short DNA fragments. In the meantime, primase makes another primer. So DNA polymerase 3 jumps back, moves forward, synthesizing another DNA fragment. Does that make sense? Do you see that? I have an illustration. I can actually kind of reenact the differences. And then guess what happens as the replication fork is moving to the left? This DNA polymerase 3 will continue jumping back, synthesizing those short fragments. And we call these short fragments Okazaki fragments. Does that make sense? So leading strand synthesis here happens in uninterrupted fashion. I'm reading a book from chapter 1 to chapter 24. On the lagging strand, synthesis happens in this Okazaki fragments fashion. I read chapter 24, then chapter 23, then chapter 22, and I have to jump through the book over and over. Does that make sense? I usually represent it this way. So imagine that I am DNA polymerase 3, and I am moving along the from 3 to 5 here leading strand. This is how I'm going to move. Right? No interruptions. I'm moving forward. Now, replication 4 goes same way, but now I'm DNA polymerase 3 on the lagging strand. Look at me. Look what I'm doing. Make a giant leap backwards and then synthesize forward. Giant leap backwards, synthesize forward. Does that make sense? I jump back, make a little progress to make DNA fragment. We clear about that? So leading strand, uninterrupted, lagging strand, Okazaki fragments. And this fork will be moving to the left. And remember, this fork, where everything else is happening, will be moving to the right. Now, very simple question. Which strand will be leading and which strand will be lagging in this fork? Use the term up or lower. Say again? Are you sure? So you suggest that on this strand, this will be a leading. This will be lagging, and the bottom will be leading. Okay? Make sense? Now, look at this picture. Let me get rid of this dashed lines here. And let me ask you the question. Look at this picture. I'm going to ask you one question that is very easy. On a scale from 1 to 0, it's 1 to 10, it's -1. During DNA replication, what are we synthesizing? DNA. Thank you very much. No, that's right. No irony. We synthesize DNA. Considering that we are trying to synthesize a complementary DNA molecule, is there any problem in this picture? Do we face any problem? Do we have an issue here? That's 3 out of 10, maybe 2 out of 10. Say again? No, everywhere. Well, mostly in lagging, yes, mostly in lagging, but in leading a little bit of a problem too. Remember, we're trying to synthesize DNA. What are those green things? Huh? No. These green, not these ones, these ones. What are they made of? It's right there. And what we're trying to synthesize. It's an issue, right? We don't need RNA in our DNA. So what are we going to do? We're going to place, I'm running out of colors. We're going to place special protein. I'm not going to draw the protein. I'm just going to show it that it's going to be placed here. The protein is called DNA Polymerase 1. So what it does, it removes RNA nucleotides and replaces them with the DNA nucleotides. Does that make sense? Got it? I don't really have a good analogy for it. Okay? But go ahead. Where? Like on the ligand strand. Yeah. So now look at this. We need to replace primers, and DNA polymerase 1 does just that. It replaces RNA primers with the DNA nucleotides. That makes sense. There is one final, well, not final, but penultimate problem. When these Okazaki fragments are formed, they are not connected to each other. Imagine that you're building a fence, and I recently discovered, I know that it's kind of embarrassing, but I recently discovered that you don't need to build a fence from individual planks. You can buy sections, Home Depot, and install them. Good to know. But imagine you buy like 10 sections, but when you install them, you have to link them. Does that make sense? You can't just put a section and put another one next to it. They have to be connected. Turns out Okazaki fragments, even after RNA is replaced by DNA, they're not connected. They're like fence elements. There's an enzyme. No, seriously, I ran out of colors. There is an enzyme called ligase, DNA ligase, that stitches those elements, fences. Does that make sense? Okay, so let's give it a run, like in a smooth manner, and we'll chat about one last feature, and we'll talk about mutations. So DNA gyrase on supercoils prevents supercoiling of DNA molecule. Helicase separates two strands, and separation starts at the origin of replication. There are a lot of AT pairs. And then single-stranded binding proteins prevent DNA strands from coming back together. DNA polymerase primase synthesizes primers for DNA polymerase 3 to start DNA synthesis. DNA synthesis happens in uninterrupted and continuous fashion on the leading strand because DNA polymerase 3 reads the DNA from 3' to 5', and the direction 3 to 5' matches the direction of replication fork movement. On the lagging strand, though, 3 to 5' direction is opposite to the replication fork movement. Therefore, DNA polymerase 3 has to synthesize DNA in short fragments that we call Okazaki fragments, jump backwards, and do this jump backwards stunt till the end of replication process. RNA primers are replaced with DNA by the enzyme called DNA polymerase 1, and then Okazaki fragments are stitched together by the enzyme called DNA ligase. Last little bit. Does anyone remember what is the shape of the chromosome in the bacteria? So when you have a circular molecule, look, and another circular, and when you copy a circular molecule, what you're going to end up with, and it's pretty interesting, you're going to have two circular molecules linked to each other, like the chains of the, like links of the chain, okay? It's called concatenated molecules. And here's what you need to do. Look at this. You need to do this. There's an enzyme called topoisomerase 4, which does exactly that. Separates concatenated molecules. Okay? So far, we're good. Now, why the hell do we need to know that? One reason, we need to understand how some biotechnological applications work, and actually knowing how DNA polymerase 3 works was a birth of the modern DNA polymerase 3, DNA ligase, was the birth of the modern recombinant technology. A lot of high-level biotech has been made possible because we know how these proteins function. Second thing, look at this. If you can inhibit some step of DNA replication process, you can essentially inhibit the reproduction of bacteria. Does that make sense? So there's a drug that inhibits DNA replication, and we're going to talk about antimicrobial drugs in the next unit, unit number 3. And I will chat about that particular drug that many of you, I bet, know pretty well. Make sense? And yeah, that's all recorded. You know, recording's going on right now, so you'll be able to rewatch it. Good? Okay, let's talk about mutations. First of all, I want to make very clear that mutations can happen only in the DNA or RNA. Okay, no exceptions. When people say mutations in the protein, that's wrong. Mutations can lead to changes in the protein, and we'll discuss it a little bit, okay? But mutations cannot be in the protein. Now, I have a question for you. It should be a simple question. What do we call a process of RNA synthesis in the cell? RNA synthesis based on the DNA template. Different transcription. So the mistakes in RNA can be introduced during transcription. Does that make sense? Quite an unusual question. Do we care? Why? Huh? Yeah, it's RNA. It's disposable. It's going to be degraded. If there's a mutation in RNA, okay, we're going to get few bad proteins. Cell will break them down and we'll start again. Does that make sense? So RNA mutations aren't, we're not talking about viruses, totally different story. Not even, I will address viruses closer to the end, okay? So what we care about is mutations in the DNA. Does that make sense? So we're going to focus entirely on the mutations in the DNA. What can cause mutations in the DNA? Say it again? No. I'm sorry, that's my English. Not what mutations can cause, but what can be the cause or changes in the DNA. Does that make sense? What can cause mistakes in the DNA? Any ideas? Excellent, thank you. So first, can be replication mistakes. What else? Sorry, no. Transcription is you use DNA template to synthesize RNA. Mistakes in RNA, sure, but template is not effective. What else? Anyone ever watched any zombie apocalypse movie or just apocalypse movie? What turns human into piles of rotting flesh? Really? Virus can cause mutations, but usually those are pretty substantial fragments. I wouldn't even, it's like massive changes. That can happen. Some viruses can do that. But more frequent. I mean, viruses became kind of an obsession in the later years. What was the main topic of like world apocalypse in like 1950s, 60s, 70s? Oh, the new generation. Nobody heard about duck and cover. Nobody put the gas masks in school. Nuclear war. So radiation. That's our mutants, right? And the third reason, anyone ever heard about the drug called thalidomide? I'm old. I'm really, really old. It was a drug developed in Europe in the 70s that was used in pregnant women to mitigate the morning sickness. And it worked marvelously, but FDA did not approve it because they were not convinced it was safe. Guess what? It was not. It led to massive teratogenic effects in newborns. Basically, newborns were born without arms and legs. So their feet and hands were attached directly to trunk. Like, no, this part was absent. Make sense? So what I'm trying to say is that another reason for mutations is chemicals. You know, we all know that people, for instance, you know what is the unofficial nickname for city of Houston? It's a cancer city because of all the oil refineries in the bay, you know, because it's such a polluted air that, you know, rates of cancer are much higher. Okay. So let's kind of figure out which one is more important. So how frequent are replication mistakes? I'll give you an approximate number. It's 1 per billion nucleotides. if you consider an average genome of E. coli being 5 million with M nucleotides, then the chance of a mistake in an E. coli genome is 0.5%. One mistake. So there's 0.5% chance that there's going to be one mistake in the entire genome copy. Does that make sense? So it's super low, super low. By the way, for RNA, the rates are much higher, and I can kind of, to digress a little bit and make it live a little bit more. That's the reason why we get vaccinated against flu every year. Because influenza has RNA genome, and when RNA gets copied, it accumulates mutations really fast. Does that make sense? And virus changes every year. Now, a lot of people express the same concern about coronavirus, which is also the virus with RNA genome. But the trick about all coronaviruses is that they have special mechanism that corrects the mistakes in the genome. That makes sense? So yeah, there are a lot of mistakes, but they have a special proofreading mechanism that corrects the mistakes. So actually coronaviruses are very, very genetically stable. We good? So we're going to basically ignore replication mistakes because they are so rare. And we're going to focus on the mutations caused by radiation and chemicals. But before we get there, we need to define what types of mutations we're talking about. So actually I'm going to use for once this textbook as sort of a seven, I guess. I'm going to use it as a should be something. Okay, included the code. Yeah, so I'm going to use it as a material to look up some nucleotides. So mutations can be divided into two large groups: point mutations and frame shift mutations. So let's talk about point mutations first. In point mutations, one nucleotide is replaced by another. So point mutations can be silent, missense, and nonsense mutations. So let's talk about an example of a silent mutation. So for example, let me show you. The amino acid leucine can be encoded by the codon CAA. Sorry, no. That's CA. CTT. We're talking about DNA. Remember, DNA. For RNA, it's going to be CUU. Does that make sense? Does that make sense? Okay. Now, let's say we replace this last T with C, and what we get is CTC. This is still a codon for leucine. Does that make sense? So you see, is this a mutation? Yes, it is. Does it change amino acid? No, it doesn't. So it's the same amino acid in the protein. Protein doesn't change. This is why it's called silent. We're good? Okay. So example of missense will be this. So isoleucine is ATT. Okay. Now imagine this T is replaced with C. We're good? So this is the codon for isoleucine. This is the codon for threonine. Does that make sense? You clear? Good? Okay. So you see why it is missense, because one amino acid becomes replaced by another amino acid in the protein structure. Does that make sense? Good? And finally, nonsense mutation. So imagine you have a codon TGG, which encodes tryptophan, but G gets replaced by A, so you get TGA, which becomes a stop codon. Does that make sense? Okay, so again, silent mutation. Amino acid doesn't change. Missense mutation. One amino acid changes to another, replaced by another. And in nonsense mutation, the codon for amino acid is replaced by stop codon. Now, I have a question. When one amino acid is replaced by another in the protein, does it necessarily change the structure of the protein? Grace is right. Not necessarily. It can, but not necessarily. Does that make sense? In this case, it most likely will. This is hydrophobic amino acid. This is hydrophilic amino acid. But in some cases, it will not. We good? Now let's talk about frame shift mutations. So I'm going to write down some codons here. To make my life easier, if you don't mind, I'm going to write down RNA instead of DNA because it's all in RNA. But the principle is the same. Now let's take a look at the frame of reading. That's your first codon, right? That's the second, third, and fourth. You see that? Got it? Now imagine for 1 second that there is a deletion. And this U is gone. Okay? Let's see what we've got here. We've got, and original one was methionine, CCU proline, phenylalanine, and valine. Now look what we get now. Frame, 3 codons, or 3 nucleotides, right? Another frame. Now look what happens to the frame of reading. Look. So now we have methionine and proline, but here we have UUG, which is, instead of phenylalanine, it's leucine. Whatever is here is going to be totally different. It's not going to be valine. It's either going to be phenylalanine or leucine as well. Does that make sense? So look what happens. The entire amino acid sequence in the protein will be altered by the frame shift mutation. And that is absolutely going to screw up the protein function. Does that make sense? So I'm just curious, if you would compare the relative effects of frame shift mutations versus point mutations, which exert more dramatic effects? Frame shift, most likely, yeah. And they can result from either deletion or insertion. So far, we're good? Okay. So now we're going to see what different causes of causes of mutations, what kind of mutations they can lead to. So chemical and radiation. We're going to start with chemical mutagens. First one will be nucleotide analogues. In this case, this is something that we as people do not have to worry about. These are used for research purposes. Okay, that makes sense? So nucleotide analogues, let's say you had a T pair, you use a nucleotide analog that can be incorporated in the DNA instead of A. I'm going to call this analog X. But this is an interesting molecule because in the next round of replication, X is going to be paired with C. Does that make sense? And then over time, what you have here is AT is replaced by GC. Does that make sense? So that's going to be point mutation. Good? Okay. I'm trying to remember all of them. Nitrite, nitrous acid. So this causes point mutations when AT pair is replaced by GC. You probably have heard, I'm not going to ask you, there's a reaction in the notes. You don't have to remember that reaction, don't worry. Okay, you probably have heard about the link of nitrite in processed meats linked to colon cancer. Right here. I mean, nobody actually proved the causative relationships, but it's quite conceivable that nitrite can increase the risk of colon cancer due to its ability to cause point mutations in the DNA. Does that make sense so far? And then we have intercalating agents. Which cause frame shift mutations. So how they do it? Now, if you look at this decrepit DNA model right here, okay, you can see those few bases right here. Can you see that? Few pairs. So what intercalating agents do, they incorporate themselves between the pairs. Got it? That changes the structure of the DNA molecule considerably. And when this DNA molecule becomes copied, that leads to either insertions or deletions. Now, intercalating agents are super useful in, brain fart. They are very useful in research because they bind to DNA very well, and you can use them to stain DNA, like acridine orange or ethidium bromide. But they are, due to their ability to cause mutations, they are extremely carcinogenic. Okay? Now, radiation. Ionizing radiation and non-ionizing radiation. So ionizing is gamma rays and X-ray. Non-ionizing is UV light. So the mechanism for ionizing radiation is double-strand breaks, and that leads to both point and frame shift mutations. Now, I want to highlight here in ionizing radiation, it's not so much as breaks themselves cause the problem, but the repair process causes the problem. The repair of double-strand breaks is imperfect because both strands are broken, there's no backup, right? That makes sense? Non-ionizing radiation, formation of thymine dimers. This is how it's going to look. Look at this. Imagine that you have a DNA strand and you have two adjacent thymines. There will be a bond formed between them. That bond will cause kinking of DNA and mistakes during the replication, so also point infection. Okay. So actually, this flowchart summarizes what you should know of the exam. Like, I'm not going to ask reactions, okay? I'm not going to ask specifics like acridine orange or ethidium bromide. Go ahead. Thymine dimers, yeah. Formation of thymine. Pyrimidine dimers? Thymines, thymine and cytosine are pyrimidines, and that's in the study guide. You need to know which nucleotides are pyrimidine nucleotides, which are purine, but thymines and cytosines are pyrimidine nucleotides. Thymine dimers is just most frequent. Cytosine, can it happen? Yes, but thymines are just, that is more frequent. Okay. So what can we do about the mutations? We can repair them. And I specifically, I will be focusing on the thymine dimer repair, okay? So UV light acts on the DNA. Okay, and remember what happens when you have two thymine residues next to each other. I just pictured it. What happens is they become chemically bound, right? That makes sense? Just mention. There are two options. Option number one, when there's light, like seriously, when light is shining on your DNA, the enzyme called photolyase will just break down this bond. And you have your intact DNA molecule. Does that make sense? That it's that simple. And then there's another option, which is called excision repair. Happens in the dark. Look. So first, the enzyme called endonuclease, it will introduce cuts. That makes sense? You see these little breaks that I pictured? Next, this fragment will be removed from DNA. The enzyme that removes the fragments is a familiar name, a helicase. Does that make sense? And then this part will be filled with another familiar name, DNA polymerase 1, and the ends will be stitched by the third familiar name, ligase. So an excision repair endonuclease cuts out the damaged fragment. Helicase removes it. DNA polymerase 1 fills up the gap, and ligase stitches the ends. Does that make sense? Those are two repair mechanisms that I want you to know. Now, last bit before we're going to take a break, going to be a conversation about the Ames test. Basically, with the Ames test, we're going to wrap up the genetics. I wanted to do it before the break because it's kind of logical. Before I start picturing something, I want to make a few remarks. So first, not trying to scare you, just letting you know. I rarely do that, but there's going to be only one question on Ames test in the exam. It's kind of a big question. Not multiple choice, but it's the hardest question on the exam. Okay? You can answer it if you listen closely what we're talking about and if you understand the Ames test. But just, I'm trying to get your almost undivided attention. Second remark is, what do we do the Ames test for? Imagine that you are a pharmaceutical company and you are testing a new drug. And you don't want the thalidomide story. You don't want your drug to be a potential mutagen. Does that make sense? So how do you figure out if it causes problems? Well, one way to do it is just take a bunch of mice, give them a drug and see if they survive or not. So, but look, if they do not survive, then yeah, it's a lousy chemical. You're not even going to move on, right? You can give it to pregnant mice and see what happens to the offspring. And if the offspring doesn't survive, yeah, that's a lousy animal too, lousy medical chemical too, right? But what if you give the drug to mice and they survive, and the offspring survives? I mean, you can take mice to the opera. They can talk to you. They can say they feel crummy, right? And especially if you're looking at mutations and maybe associated tumors. How are you going to look for a tumor in a mouse? Where? What size of a tumor? When? Does that make sense? It really, really, really complicates things. So in order to address whether a chemical has a potential for mutagenesis, we do Ames Test, which we do on bacteria. And I'm going to break down the whole idea of the Ames Test, hopefully in a rather simple manner. To avoid confusion, I'm going to be using the same example that you have in your lecture notes. Okay? So basically, I'm going to walk you through the lecture notes. We're going to walk through that together because this is kind of conceptually a little bit harder than usual. So we start with a agar plate with the salmonella on it. And this is a defined complete medium. Does anyone remember what defined medium means? What do we know about defined medium? We know its chemical composition. So we know exactly how much amino acids, vitamins, glucose we added to the medium. Does that make sense? We know exact chemical composition, and our salmonella grows marvelously on that medium. And then, now, this is step number one, which is really important. We take two empty plates. These plates are different from one from another. This plate, look, the top plate is same defined complete medium. And the bottom plate, same medium, but without one amino acid, without amino acid histidine. So we're going to say that this is histidine negative medium. Does that make sense? And this, we're going to call it histidine positive. Okay? Now look at this. I told you before that this medium and this medium are the same. So what you do, you take a little velvet stamp, put the stamp on the colonies, transfer them on this medium. Okay? What you can expect exactly the same pattern of growth. And then you use the same stamp, the same source, same colonies being stamped on this medium. And here's what you see. Look. Is there a difference? One colony doesn't grow on the histidine negative plate. 3 out of 10 question. Why? Why bacteria in that colony can grow on the medium with histidine, but cannot grow on the medium without histidine? Say again? Yeah, absolutely. Because I took the amino acid out and they need it and they cannot make it. You see the difference? These 5 colonies, they can make their own histidine. This colony right there, cannot make its own histidine. Does that make sense? So we have, now, I'm going to ask you a series of questions which are about 2 out of 10. Is inability to make histidine, is it a phenotype? Yes, absolutely. It's a phenotype. Inability to make histidine. What determines a phenotype? Genotype, genes expressed at a specific period of time, right? So therefore, there's some genetic differences between this colony and this colony. Most importantly, we can map those genetic differences to the genes responsible for histidine synthesis. Does that make sense? So we can picture it this way. Look, if this is the DNA of a wild type, this colony, right? Then this is the DNA of a mutant. And look, I'm trying to color code it. There is a mutation that makes mutant unable to produce histidine. Does that make sense? Now, 2 out of 10. Mutant that cannot produce histidine. Can it, generally speaking, still grow? What does it need to grow? Histidine. Microorganisms that require a particular nutrient or a growth factor are called auxotrophic. A good example of auxotrophic organisms, there are 8 auxotrophic organisms that are currently in this room. That's us. We need essential amino acids. We cannot grow without essential amino acids. Does that make sense? We are auxotrophs. So far, we good? Okay, so that's a histidine auxotroph. Do you follow the wild type and mutant with the mutation in the histidine locus? Now, it will be, it's not a leap of logic. You just have to understand. So what we do, we take this mutant, and I'm going to represent it as a DNA. We're going to take this mutant, and we're going to expose it to our chemical. Okay? Now let me ask you this. If your chemical is absolutely benign, non-mutagenic, ideal case scenario, is it going to change the DNA? No, everything's going to be the same. The phenotype of that microorganism will remain the same. But imagine that your chemical is mutagenic. It will incorporate mutations all over the DNA. Does that make sense? And what's most important, it may mutate this mutated part and revert it back to wild type. Does that make sense? Like double negative gives you positive. A mutated mutation gives you a wild type, reverts to the wild type. Does that make sense? Got it? Now the key point, and I want you to work with me to kind of go back and forth. This is an agar plate of the histidine negative medium. Histidine negative. If your chemical is absolutely benign, non-mutagenic, all that kind of stuff, will your bacteria, after exposure to absolutely benign, non-mutagenic chemical, will your bacteria be able to grow in histidine negative medium? Why? You're right, absolutely right. Why? There's nothing changing, right? If your chemical is mutagenic, will bacteria be able to grow? Yes, because some of those cells will revert to the wild type, and this is what you're going to see. Does that make sense? okay? So basically the idea is you identify a microorganism which has a mutation in the DNA, which gives it a particular phenotype. And then you see, and you need to link that particular phenotype to a particular part of the gene, a mutation in the gene. And then you see if you can expose this mutant to a mutagen and revert it to wild type. So for instance, if you would treat your mutant with a control, I don't know, a buffer, no chemical, would you have any growth? No, because there's no reversion. Does that make sense? Now, in the lecture notes, there are only two samples, like a chemical and a control, right? I can think of 3 or 4 more controls to put there to make the experiment better. And also they talk about the red liver lysate. You may have seen that there. Red liver extract. They use it in the lecture notes. The liver extract is often added here to see if metabolites of your chemical can be mutagenic. Does that make sense? So not the chemical itself, but the metabolite of the chemical. Got it? Because the liver may change it. And we have a lot of prodrugs like that, actually. I was reading about some drug, I can't remember from top of my head right away, but oh yeah, the drug that I think it's 5-piravir. Yeah, 5-piravir that is being studied for potential inhibition of coronavirus replication. The drug itself is a prodrug because it gets modified in the body. So that's what liver extract is. We good? Let's take a break, and when we will come back, I will get out your plates. You will take a look at them, and we're going to do the lab. Okay, so we're going to talk about some aspects of microbial metabolism. Most of the conversation we're going to hold next week. Okay, it's going to be your beloved glycolysis, Krebs cycle, oxidative phosphorylation, etc. Today I want to talk about metabolic strategies that microbes can utilize, the concept of oxidation and reduction, which will come in the next week, and enzymes. And that'll be it. So, metabolic strategies. 2 things that, generally speaking, microorganisms need to grow are energy and carbon. Now, energy is kind of obvious, right? You need energy to grow. Why carbon? All organic molecules contain carbon, so you need carbon. Now, where can you take energy from? You can take it from the sun. In this case, you are called phototroph. Or you can take it from a chemical source. In this case, you're called chemotroph. Example of chemotroph will be garden variety of bacteria, and example of phototroph would be photosynthetic microorganisms such as cyanobacteria or microscopic culture. Now, what about carbon? 2 sources for carbon can be organic source, right? And that example of organic carbon will be, for instance, glucose or methane. Okay. Those are examples, not necessarily. That's not the only source. The point is organic carbon, or generally speaking, any dead organisms. Okay. And then there can be inorganic carbon as a source. Okay. For instance, carbon dioxide or carbonate. Okay, now, organisms that consume organic carbon, basically we can say organisms that consume other organisms are heterotrophs. Organisms that can convert inorganic carbon into organic form are going to be autotrophs. Does that make sense so far? Good. Now look what happens when we characterize a microorganism based on its energy source and carbon source. Take bacteria that you are growing on your plates. Where do they take energy from? Take it from the nutrients in the plate. So they are chemotrophs. Where do they take carbon from? They take it from the organic compounds on the plate. So they are heterotrophs. So we're going to call them chemo heterotrophs. Take cyanobacteria that use sunlight as the source of energy. And these cyanobacteria can run a photosynthesis reaction, which allows them to convert CO2 into carbohydrates. So they become autotrophs. Now, there are microorganisms, different sulfur bacteria, that can use energy of sunlight as the energy source, but will require organic carbon. They will be photoheterotrophs. Does that make sense? And finally, probably the most interesting organisms, the ones that use chemical sources of energy that can convert inorganic carbon into its organic form, chemo autotrophs. We're not going to be talking about these chemo autotrophic bacteria or archaea, but they are extremely interesting. Those are the microorganisms that are usually found near the deep sea high temperature vents, volcanic vents. You know what I'm talking about? Like when the temperatures are extremely high, there's very little organic matter, therefore autotrophs. There's plenty of inorganic carbon, more than you can imagine. There's no sunlight, so they cannot use sunlight. So they have to use chemical sources of energy, which are plentiful, and they have to convert inorganic carbon into organic compounds. They can survive at extreme temperatures, extreme pressures, extreme pH. Really interesting microorganisms. Okay? So far, we're good? Now, the entire sort of compendium of reactions, chemical reactions in a microorganism, as you may have learned from the notes, is called metabolism. So all reactions can be broadly divided into reactions of synthesis, like reaction of DNA synthesis, and reactions of synthesis are called anabolic. And reactions of breakdown, which are called catabolic. So for instance, the breakdown of starch by some fungi will be catabolic reaction, while synthesis of proteins by those same fungi will be anabolic reaction. Usually, anabolic reactions require the input of energy. And usually, again, not always, but usually, catabolic reactions produce energy. So reactions that require input of energy are called endergonic. and reactions that produce energy are called exergal. So in this case, synthesis of ATP is anabolic endergonic reaction, and breakdown of ATP is catabolic exergal. We good so far? Okay, great. Now, I want to talk about oxidation and reduction, and it's going to be more of a terminological conversation rather than heavy chemistry conversation. So let's take a look at two reagents, A and B. Okay. I'm not very much interested in the products, but I'm interested in what's going on here. So A, reagent A, loses its electron to reagent B. Okay, so we can write down that A loses electron and B gains electron. Okay, good so far? Okay. Now, by definition, when A loses electron, it is oxidized. Now, let me ask you this. If A, when loses an electron, is oxidized, what oxidizes A? The answer is right there on the board. A loses an electron, it is oxidized by what? No. It loses an electron to what? So if A is oxidized, it's oxidized by B. Yeah. My favorite example, imagine that, I don't know, somebody pays me for service. So that person pays me, but I am being paid by Lakeland Community College pays me salary. I am being paid by Lakeland Community College. Does that make sense? So A is oxidized by B, therefore B oxidizes A. Does that make sense? Now B is reduced by what? A, so therefore A reduces B. Okay, so in this situation, since A reduces, it is called a reductant. And B is called oxidant. Does that idea make sense? We good so far? Now I want to say a few words about the whole process of metabolism and getting energy out of chemicals in the cell. There are two most common approaches to the conversation about the energy flow in the cell. So first of all, let's describe what energy flow I am talking about. So we've got nutrients, right? Now nutrients have to be broken down to extract energy to make ATP, and then ATP is used in the cellular processes. Does that make sense? That makes sense, what I just described. Got it? It's pretty straightforward. Now, there are two approaches. You cannot, due to the mask, you cannot see that I have a sarcastic grin in my face, but the two approaches, the biology approach and the chemistry approach, which are also the wrong and the right approach. Biology approach is wrong, chemistry approach is right approach. Since I've got my bachelor's degrees in chemistry, that cannot be. Just chemistry approach is the only way. Now, some folks in our department, they use biology approach. With all due respect, it's a wrong one. I'm not very much concerned about being beat up because Dr. Lendis is teaching online. And I also have a really powerful, literally really powerful backup. If you ever saw Dr. Jason Thomas teaches chemistry, you can imagine that having a protector like this gives me some additional bonuses. Now, speaking seriously, biology approach uses the idea of transfer of hydrogen ions, atoms from one chemical to another, which from chemical standpoint makes no sense. Because the energy flow in the cell is about breaking down the bonds and forming the bonds, oxidating and reducing. Does that make sense? So since oxidation and reduction is about the electron transfer, we're going to be discussing this approach from the standpoint of electron transfer. And broadly speaking, this process right here, specifically, we're going to focus, look, we're going to focus on this one. This process can be described in a very simple schematic way. So if this is energy of electrons, we have a molecule with a really high energy electrons, okay, basically electron donor, and we have a molecule with a low energy of electrons, the acceptor, and electrons have to go from donor to acceptor. Does that make sense? Think of it this way. There's a boulder right there on the roof. It has certain potential energy. When you throw it off the roof, by the time it reaches the ground, it has a kinetic energy. Does that make sense? So these electrons, when they will be moving from donor to acceptor, they will give off that much energy. Does that make sense? And that energy will then be converted into ATP, energy of chemical bonds in ATP. So it's all about the transfer of electrons. Does that make sense? Now imagine that, imagine that your boulder on the roof, the electrons, they aren't just falling off the roof, they have a pretty convoluted path, okay? With a lot of twists and turns. Basically, what I'm trying to say is that in the cell, one reaction can happen, quote unquote, in this corner of the cell, another reaction can happen in this corner of the cell. Does that make sense? Electrons cannot exist by themselves. So they have to be transported, they have to be carried from one part of the cell to another, from one molecule to another. Is that clear? How do we do it? We use electron carriers, as I sometimes call them, the electron Ubers. Because basically what electron carriers do, they accept electrons and then they unload them somewhere else. 3 electron carriers that you must know are NAD+, FAD, and NADPH. This last one, sorry, NADP+, not NADPH, NADP+. This last one, we will encounter it when we're going to talk about photosynthesis, okay? Now, these electron carriers will gain 2 electrons. Electrons are negatively charged, So to balance electrons, you need something positive. That will be hydrogen atoms, hydrogen ions. Does that make sense? You see that? So two positives counteract two negatives, okay? This is where this wrong perception of biologists is coming from. It's not a transfer. Hydrogens are just for the balance, okay? Does that make sense? And what you end up with is NADH and balancing one, FADH2, and so before we move any further, I want to make a quick note here. When we're going to be talking about the reactions in the cell, reactions in the Krebs cycle, reactions in the glycolysis transition step, just, I'm not going to be writing these hydrogens if you don't mind. Like, we kind of know that they're there just to minimize the writing, okay? Kind of, asking for your permission, sort of. Okay, we're good? Okay. Now, look at what's written here. Look at what's happening here and tell me, when these electron carriers gain electrons, they are what? Reduced. So these are reduced forms. Now imagine that you are running this reaction backwards. When these electron carriers bring the electrons to the destination point and unload them, when they unload them, when they lose them, what happens to them? They are oxidized. These are called oxidized forms. Does it make sense? Good, clear? So that was kind of, important conversation about reduced oxidase. Now we're gonna, quickly glance over the concept of the enzymes, without which the metabolism in the cell is impossible. So enzymes. This is how enzymatic reaction can look like. So this is the enzyme, okay? This is a substrate of an enzyme. Substrate binds into the active site of an enzyme. Enzyme does its enzymatic magic. And what you end up with is the enzyme and the product. Do you appreciate that product and the substrate are different? Do you see that? Okay, that's important. Now, what can we say? Now, why enzymes? What are enzymes? Enzymes are chemical catalysts. So what they do, they increase the rate of reaction. Okay. They do not change the outcome. They do not change the yield or ratio of products. Think of an enzyme as a car. Anyone who lives relatively close here? I don't need to know where. Relatively close. Relatively close. You probably can walk to Lakeland. It'll take you a decent amount of time, right? Or you can drive to Lakeland and you're going to get here much faster. Outcome is the same. Your car, in this case, is a catalyst of the process. It makes it faster. And look, few other features come out of this. First of all, since you have the keys, only you can drive the car. So enzymes are extremely specific. Okay, When you arrive here, the car remains the car. Nothing changes, right? Like it's not falling apart or disappears. So enzymes are not wasted in the reaction. Does that make sense? And finally, they have optimal conditions. Which is important. I illustrated using an example of DNA polymerase that we've already learned about. So if you take DNA polymerase and measure its activity, as it depends from the concentration of magnesium ions, it's not hypothetical. That's more or less how it happens. Look at this. Its activity will change in kind of this way. When there is no magnesium, it's not working terribly well. There is too much magnesium, it's not working terribly well. There is some optimum right here. Does that make sense? We're clear. So have an optimal concentration. You can apply to pH, to the temperature, anything. For instance, DNA polymerase that is used in PCR, remember we talked about PCR? Its optimal temperature is about 85 degrees. But we run the reaction at 72. It still works at 72 all right. That makes sense. Okay, awesome. Now, how enzymes increase the rate of reaction? So if this is a graph of energy versus time, let's say that's the energy of your reagents. And that is the energy of your products. The reaction is not going to go like a smooth ride down here. It will need to go over the pretty large hump, which is called, that hump right here, it's called activation energy. A good comparison to this, remember our boulder on the roof example? If you have a pretty, a boulder sitting on the roof, you can't just drop it down. You need to get it over the railings. Does that make sense? So your boulder can have enormous amount of energy. It will definitely fall by itself. But first, you need to get it over the railings. These are railings, the activation energy. So what enzymes do, they lower the railings. So the reaction goes faster. They lower the activation energy. Does that make sense to you? Now, a few words about the couple of types of enzymes. So first of all, there are endo and exoenzymes. It's all in the lecture, just wanted to highlight a couple of features. Enzymes that remain in the cell are endoenzymes. For instance, today, where we're talking about DNA replication. DNA replication happens inside of the cell. All the enzymes that we have discussed, DNA ligase, helicase, DNA polymerase 3, blah, 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 all those enzymes are endoenzymes. Now, if you remember, a couple of weeks ago, we were talking about the bacterium called Staphylococcus. Remember that? And I mentioned the condition called scalded skin syndrome. When superficial layer, the epidermal layer of the skin peels off. Remember that? It peels off because the enzyme, that's the Staphylococcus, producing, is released from the cell and it breaks down the basal membrane. Does that make sense? So that's going to be an example of exoenzymes. From the standpoint of pathogenesis, from the standpoint of what bacteria do to human body, exoenzymes will be of primary interest. Does that make sense? Like collagenase of Clostridium perfringens that is released from the cell into the connective tissue, breaks down collagen. We're clear. Enzymes also can be constitutive and regulated. Constitutive enzymes are produced at the same level. They are rare. Regulated enzymes, well, are regulated. And I want to kind of demonstrate how enzymes can be regulated. So we're going to talk about inhibition of enzymes. There is activation. I'm going to say a few words about activation, okay? But those are going to be words, not so much pictures. So inhibition, competitive, versus non-competitive. In competitive inhibition, inhibitor binds to the active site. So for instance, if Kenna is a substrate, I am the inhibitor, and the chair is the active site, we're going to compete for the chair. So right now she wins. That makes sense? But like if I sit there first, that's it. She cannot take it. Does that make sense? Now in non-competitive inhibition, inhibitor binds to some other site on the enzyme, not to the active site, but it changes the shape of the active site so the substrate cannot bind. So using that analogy that we just used before, look at this. So Kenna is sitting on a chair. Imagine that that empty chair next to her is that other side. Let's say if I'm stinking really bad, if I was like covered in skunk, if I will sit on that chair, she will not occupy that one. That makes sense? Because I'm somewhere else and I change the properties of the active site. You got it? So since in this case, inhibitor binds to some other site, non-competitive inhibition is sometimes called allosteric. Allo means different. Steric means space. Okay, so allosteric inhibition and non-competitive inhibition are synonyms. Does that make sense here? As you can imagine, it can be also allosteric activation. There's a molecule that binds to some locale on the enzyme and activates. Okay, we good? Okay. Last little bit, well, a couple of bits about the enzymes. First, the idea of holoenzyme and apoenzyme. It's pretty straightforward. So imagine you have a protein, an enzyme. Look, there's an enzyme. It's a protein. And it's dysfunctional, okay? It doesn't work, okay? And then there's a cofactor or coenzyme. It's usually a small organic molecule. Can be an ion, like magnesium or molybdenum or chromium. Only when these two come together, they will form a functional unit, which is called holoenzyme. Does it make sense? So apoenzyme has to bind to a cofactor to form a functional holoenzyme. Okay. Now the very, very, very last piece of the enzymes. The idea of enzymatic partners. Since we're going to be essentially talking about various metabolic pathways, enzymatic pathways. Enzymes are rarely working by themselves, like one enzyme. They usually are parts of the bigger metabolic pathways, such as, for instance, you can have metabolic pathway that is linear. A, B, C, D. That makes sense? You can have metabolic pathway that A, B, C, D can be divergent. You can have a metabolic pathway that is a cycle. Does that make sense? All of those are examples of metabolic pathways. And what I want to highlight, and A, B, C, D, all these letters, they represent certain products, okay? Certain intermediates. What I want to make very clear is that sometimes a product of a pathway can serve as the inhibitor of the pathway itself. So imagine that this linear pathway runs and it goes on and on and on, and the product E accumulates. Are you following me? And if there's too much, this product can serve as an inhibitor to, for instance, this enzyme or this enzyme. Does that make sense? So organization of enzymes in the pathways with the ability of some of the products to serve as inhibitors, competitive or non-competitive, okay, it is extremely valuable for the regulation of enzymatic processes and stuff. Good? Any questions? Okay. I'm gonna wrap it up.